Every second, about 2 people die on Earth, 108,000 a day, almost 40 million a year. This is the only event that is absolutely guaranteed for everyone reading these words, and yet it is the least studied of everything that happens to us. Most people know about death exactly as much as fear allows. Try not to think about it. But the science of the last 20 years has overturned almost everything that was considered obvious. The border between the living and the dead turned out to be not a line, but a zone. The brain of a dying person in the final seconds works more actively than in a state of wakefulness, and people who were resuscitated after cardiac arrest described the same thing, regardless of country, language, or religion. Ahead lie the physiology, neurobiology, and biochemistry of death, without mysticism, but with facts that are unsettling. Chapter 1: 4 minutes. In 1967, American cardiologist Michael Reeser recorded a case that was subsequently cited in textbooks for decades. A patient was admitted with cardiac arrest. Resuscitation took 23 minutes. The heart was started. The person survived. But he could no longer speak. Irreversible brain damage. 23 minutes without blood flow turned out to be too long a period. Back then, it seemed obvious. The brain is more sensitive to oxygen deprivation than any other organ. Neurons begin to die as early as 4 to 6 minutes after the cessation of circulation. That is precisely why in emergency medicine protocols, 4 minutes is the boundary. Before it, resuscitation has a high probability of returning a person without losses. After it, the chances of full recovery drop sharply. But what exactly happens inside during these 4 minutes? This is not an instantaneous shutdown. It is a cascade. In the first seconds after the heart stops, the brain still works on residual oxygen stored in the blood. Consciousness is maintained for 5 to 15 seconds, which is why some people, losing consciousness from a heart attack, manage to realize what is happening to them. Then darkness comes. In the second or third minute, cellular pumps begin to fail. Neurons maintain a potential difference between the internal and external environment. This is the basis of the brain's electrical activity. This requires energy in the form of adenosine triphosphate. Without oxygen, its production stops. The pumps stop. Sodium and calcium ions rush inside the cells, and potassium flows out. The electrochemical balance collapses. In the 4th to 6th minute, what neurobiologists call excitotoxicity begins. Neurons in agony begin to massively release glutamate, the brain's main excitatory neurotransmitter. Neighboring cells receive an avalanche of signals and become overloaded. This triggers a chain reaction of death. Cells literally burn up from the inside from overexcitation. By the 10th minute without resuscitation, the irreversible death of neurons begins in the hippocampus, the structure responsible for memory and orientation. That is why survivors of prolonged clinical death often lose memories or acquire cognitive impairments. But here's the paradox that medicine discovered relatively recently. Death is not a moment. Death is a process, and it is reversible for much longer than was thought. In 2019, a group of scientists from Yale University, under the leadership of Nenad Sestin, conducted an experiment that caused a shock in the scientific community. They took the brains of pigs slaughtered at an abattoir 4 hours prior and connected them to an artificial circulation system, a device they called BrainX. The system pumped a special solution mimicking blood through the brain. 6 hours later, the brain came to life, partially. Cells began to consume oxygen. Metabolism was restored. Synapses could again conduct electrical signals. The brain did not think. There was no consciousness. But it was alive 4 hours after the death of the animal. This study overturned the understanding of the irreversibility of death. It turned out that cellular death is not an instantaneous break, but a slow fading that, under certain conditions, can be stopped or even reversed. In medical practice, this is already being used. The method is called therapeutic hypothermia. After cardiac arrest, the patient's body is intentionally cooled to 32 to 34 degrees Celsius. The cold slows down the metabolism of neurons, reduces the need for oxygen, and inhibits excitotoxicity. This gives doctors more time. In a number of cases, it has been possible to fully restore brain function in people who spent 20, 30, and even 40 minutes without circulation. Records in this field sound implausible. In 2015, in a Norwegian hospital in Oslo, a woman was resuscitated after 97 minutes of cardiac arrest. She survived and recovered almost completely. The body was so overcooled, the temperature had dropped to 22 degrees, that the metabolism had effectively frozen. This calls into question the very concept of the moment of death. Where exactly is the boundary? When is a person finally dead? Modern medicine distinguishes three states: clinical death, the cessation of heart and breathing, but with potentially reversible changes; brain death, the complete and irreversible cessation of all brain functions, including the stem, which controls breathing; and biological death, the irreversible breakdown of cells. Between the first and the third, several hours can pass, and it is precisely in this interval that things occur which science does not yet know how to explain. Because among those returned from the first frontier, there are people with stories that do not fit into any neurobiological model. Chapter 2. Those who return. In the autumn of 2008, in 15 hospitals in Great Britain and the United States, small tablets with images appeared on shelves near the ceilings of resuscitation rooms. The pictures were facing upward, so that a person lying on a gurney could not see them. Only someone looking from the top down could see them. This was the beginning of a study that in medical literature received the name Awareness During Cardiac Arrest. In the English version, it is shortened to 4 letters, AWARE, that are pronounced with caution in scientific circles. The project was led by British cardiologist Sam Parnia from the University of Southampton. His task was simple and impossible at the same time: to check whether people in a state of clinical death can truly perceive what is happening around them. The logic of the experiment was as follows. If a person whose heart has stopped describes how they are looking at their body from above, this could be explained as a hallucination, the work of a dying brain, or residual memories. But if they name the picture on the shelf, which they could not physically see, the explanation becomes significantly more difficult. The first phase of the study covered 367 patients who suffered cardiac arrest. Not one named the image on the shelf accurately, partly because most resuscitations took place in rooms other than where the tablets were placed. But among the participants, one case was found that Parnia describes as key. The patient, a man of 57 years, told how he watched his resuscitation from above. He described a nurse who climbed onto a special stand to perform chest compressions. He described how another medic left for equipment. He named details of the people's arrangement in the room. All of this was checked and confirmed by the staff. At the moment when the patient saw this, his brain should not have been working. The second phase, a study known as Awareness During Cardiac Arrest, Stage 2, which concluded in 2020, already covered 900 cases in 26 hospitals in several countries. The results turned out to be simultaneously more statistically significant and more uncomfortable for science. About 9% of survivors after clinical death reported some kind of experience that they could not explain, and which did not correspond to ordinary hallucinations. Of those who could be interviewed in detail during the first 24 hours after resuscitation, before the memories were erased or suppressed, every third person described similar elements. The similarity, that is what strikes researchers the most. People who have never heard of each other talk about the same thing. The sensation of leaving the body and looking from above, including the description of details that are subsequently confirmed. The sensation of moving through a dark space or a tunnel. The experience of a bright, non-glaring light. Meeting people, most often deceased relatives. And the main thing, what most describe as the most important element, a review of their own life. Not a memory, specifically a review, as if the entire lived life unfolds simultaneously, with the sensation of experiencing every moment anew, including how their actions affected other people. Neurobiology offers several explanations. Hypothesis 1. All of this is a product of the dying brain. Hypoxia causes hallucinations. Depolarization of neurons provokes bursts of activity in zones associated with memory and self-perception. The tunnel is the result of impaired circulation in the visual cortex, which loses peripheral vision before central vision. Meetings with the deceased are the activation of areas storing emotional memory. This explanation is logical, but it has a problem. The brains of patients with a recorded flat electroencephalogram, that is, with a complete absence of electrical activity, should not generate any experience at all. Yet it is in such patients that the descriptions turn out to be the most distinct and the most verifiable. This contradiction is not yet resolved. Neurophysiologist Pim van Lommel from Arnhem, Netherlands, published one of the largest prospective studies on this topic in the journal The Lancet in 2001. 344 patients who underwent clinical death. 123 of them, 18%, reported a conscious experience. Van Lommel insists, consciousness is not an exclusive product of the brain. The brain is a receiver, not a source. The scientific community treats this idea with caution. Most neurobiologists reject it, but facts that do not fit into the habitual model continue to accumulate. And in 2023, something happened that forced people to talk about this with different arguments. Chapter 3. The Flash. On February 22, 2023, in the journal Proceedings of the National Academy of Sciences, one of the most authoritative peer-reviewed publications in the world, an article appeared which its authors named cautiously and academically. The title translated roughly to Neural Correlates of the Dying Brain in Humans. But in the media space, it was named differently, the study of the flash of death. A group of scientists under the leadership of neurophysiologist Jimo Borjigin from the University of Michigan studied the electroencephalography recordings of 4 patients who died in intensive care after being disconnected from life support systems. All 4 were in an irreversible coma. All were terminally ill. The families had given consent to terminate resuscitation measures. This allowed, for the first time in history, the recording of the brain's electrical activity at the very moment of a person's dying. Not in experiments on animals, not in simulated conditions, but in the real death of real people. What the recording showed did not coincide with any existing model. In the first seconds after the heart stopped, when blood flow in the brain ceased, the electroencephalogram of 2 of the 4 patients showed a sharp jump in activity, not a fading, a surge. Moreover, not random noise, but organized high-frequency waves in the range of gamma rhythms, 30 to 100 Hz. The gamma rhythm is that same range associated with higher cognitive functions, attention, perception, the integration of information from different zones of the brain. It is active when a person solves a complex problem, experiences a vivid emotion, or, as was shown in studies in 2008, when a person remembers significant events of their life. The flash lasted from a few seconds to several minutes. It arose specifically in the zones responsible for visual perception, memory, and self-awareness, in the occipital and temporoparietal cortex, in structures associated with what neurobiologists call the hot zone of consciousness. Borjigin was cautious in interpretation. She wrote directly, "The study does not prove that the dying experience a conscious experience, but it proves that the brain at the moment of dying is active differently than was assumed, and that this activity is neurobiologically compatible with what survivors of clinical death describe." This is an important distinction, not proof of consciousness after death, but not a refutation of the possibility either. The backstory of this work goes back to 2013, when the same Borjigin published experiments on rats. Cardiac arrest was induced in animals under anesthesia, and the brain's electrical activity was recorded. In the first 30 seconds after clinical death, the same gamma waves were recorded in all rats, organized, high intensity, in zones responsible for the integration of sensory information. Back then, many were skeptical. Rats are not humans, and we have no way of knowing what they experienced. The 2023 study closed this argument. Now we have data on humans, and it points in the same direction. Neurobiologist Christoph Koch from the Allen Institute for Brain Science, one of the world's leading specialists in neural correlates of consciousness, commented on this study as follows. The data do not explain whether there is consciousness after death, but they show that the border between life and death is structured more complexly than was thought. What exactly the brain experiences in these final seconds is unknown, but now scientists have a physiological mechanism that could explain the life review. An instantaneous, intense activation of memory and self-awareness at the moment when oxygen finally runs out. This is not mysticism. This is chemistry and electricity. But the body at this moment is already starting another process, slow, unstoppable, and in some sense stranger than anything that happens in the brain. Chapter 4. You become an ecosystem. In 2011, on the territory of a nature reserve in the American state of Tennessee, something happened that entomologists and forensic biologists subsequently cited in scientific papers for several years in a row. Researchers from the Anthropological Research Center at the University of Tennessee, colloquially this place is called the Body Farm, recorded the exact moment when a dead human body ceases to be a body and becomes a habitat. Not metaphorically, literally. Within 48 hours after death, 247 species of insects, 38 species of bacteria, and 8 species of fungi were identified around one body, organisms that were not here before the person died. The body pulled them in like a magnet. 4 weeks later, in its place remained a fertilized patch of soil with an altered chemical composition, in which plants sprouted that had not been encountered on this plot before. The death of one person created a new ecosystem. To understand how this happens, one must return to the very beginning, to the first minutes after the last heartbeat. While the brain experiences its final surges of activity, the body launches another process, autolysis. This literally means self-digestion. The body's cells contain lysosomes, intracellular vesicles with digestive enzymes. During life, they are strictly isolated and break down only what they are supposed to: waste proteins, bacteria, cellular debris. But the cell membrane maintains its structure through energy. When oxygen stops arriving, the energy runs out, the membranes lose integrity, and the lysosomes burst open. Enzymes flow inside the cell begins to digest itself. The first to feel this are the cells with the highest metabolism: brain neurons, pancreas cells, intestinal mucosa cells. The intestine is especially important, and here is why. In a living person, the intestine contains about 38 trillion bacterial cells. That is roughly as many as all the cells of the organism itself. They live in symbiosis. They help digest food, synthesize vitamins, train the immune system, and produce neurotransmitters. During life, the immune system keeps them strictly on its territory, in the lumen of the gut, not allowing them to penetrate into tissues. Death removes this barrier. During the first 2 to 3 hours after the heart stops, immune cells cease to work. The intestinal walls, which during life were renewed every 4 to 5 days, and were one of the organism's most dynamic barriers, begin to break down. Bacteria go beyond the limits of the gut and begin what microbiologists call post-mortem translocation, migration through the tissues. They move along lymphatic pathways and the remains of blood flow. They reach the liver in 4 to 6 hours, the lungs in 6 to 8. By the end of the first day, bacteria are evenly distributed throughout the whole body. This is not a disease or an infection, it is the return of a dead. All its life, the organism fed them, protected them, and regulated their composition. Now they process it, and they do this with striking efficiency. Parallel to autolysis and bacterial expansion, putrefaction is launched. This is a more complex process than it seems from the name. Bacteria break down proteins into amino acids, and amino acids into simpler compounds: ammonia, hydrogen sulfide, carbon dioxide, and a whole library of organic molecules with terrifying names: putrescine, cadaverine, scatol, indole. It is they that give the characteristic smell. But for a biochemist, this smell is information. By the composition of volatile compounds released by the body, one can determine with accuracy down to several hours how much time has passed since the moment of death. Forensic entomologists use this data together with the analysis of insects to establish the time of death in forensics. Insects are a separate chapter in this story. The first to arrive are blowflies, as a rule, representatives of the genus Calliphora or Lucilia. They are capable of sensing the smell of death from a distance of several kilometers and arrive within the first minutes or hours after death, depending on the temperature and accessibility of the body. A female fly lays from 150 to 300 eggs at a time, predominantly in natural openings: eyes, nose, mouth, wounds. Larvae hatch from the eggs. They feed on soft tissues and secrete enzymes that accelerate decomposition. Larvae of one generation of blowfly can eat up to 60% of the body's soft tissues in a week. Then they pupate, hatch, fly away, and their place is taken by other species, each of which specializes in a certain stage of decomposition. This is called the succession of carrion insects, a strict, predictable sequence of species that replace each other as the body's composition changes. Forensic entomologists know this sequence by heart, by which species larvae are present in the body and at what stage of development. The chronology of death can be reconstructed with accuracy down to a day. Approximately on the third or fourth day, what biologists call active decay begins. The body bloats. Bacteria produce a colossal amount of gases: methane, hydrogen sulfide, carbon dioxide. Pressure inside builds up. The skin changes color. First, a greenish tint appears in the abdominal area, where intestinal bacteria are closest to the surface. Then it spreads to the chest, shoulders, and limbs. Vessels under the skin darken. This is called marbling, one of the classic signs of decomposition, known to pathologists since the 19th century. At this stage, the body becomes maximally attractive to insects and vertebrate scavengers, crows, foxes, and in some regions of the planet, vultures, which are capable of sensing a decomposing body from a height of several thousand meters. Then, active decay is replaced by late decay. Soft tissues disappear. Skin, cartilage, and bones remain. At this stage, beetles become the main processors, especially skin beetles of the genus Dermestes and darkling beetles. They specialize specifically on dry organic remains and are capable of completely skeletonizing a body in a few weeks. The entire process from death to the complete decomposition of soft tissues takes, depending on temperature, humidity, access to air, and the presence of scavengers, from 2 weeks to several months. In the hot, humid climate of the tropics, faster. In cold and dry conditions, significantly slower. Frozen bodies in Arctic soil can be preserved for millennia, hence the mammoths and perfectly preserved bodies from the peat bogs of northern Europe, whose skin, stomach contents, and even tattoos have reached us across 2,000 to 3,000 years. Burial changes the kinetics, but not the essence of the process. Without oxygen, aerobic bacteria give way to anaerobic ones, those who live without air. Decomposition continues, just more slowly and with a different cast of participants. In a wooden coffin, a body completely decomposes in 10 to 15 years depending on the soil type. In a metal coffin, hermetically sealed, the process slows down but does not stop. Internal bacteria do their work even without external help. Cremation is the only way to radically interrupt this process. At a temperature from 850 to 1,100 degrees Celsius, organic matter burns up in 1 to 3 hours. About 2 to 3 kilograms of mineralized bone fragments remain, what is commonly called ashes, although this is, strictly speaking, not ash but calcined bone remains, which are then mechanically crushed. But here is what is important to understand. The atoms of which a human body consists do not disappear. They are redistributed. Carbon goes into the atmosphere in the form of carbon dioxide, and in a few years will end up in the leaves of trees or in the body of another living being. Nitrogen is absorbed by the soil and will become fertilizer for grasses. The calcium of bones slowly dissolves in groundwater and ultimately ends up in rivers, seas, in the shells of mollusks, or in the bones of fish. A study conducted in 2017 by Canadian biochemists from the University of British Columbia showed, in a radius of 3 meters from a decomposed body, the soil is enriched with nitrogen to such an extent that the composition of the plant community on this plot changes for several decades ahead. A person who is no longer here continues to influence the life around them, literally, through the chemical composition of the earth in which they lie. There is something in this that is difficult to talk about in strictly scientific terms, not because it requires mysticism, but because science itself here reaches questions that were formulated long before it. Death does not switch off life, it redistributes it. The brain fades, but gives off a flash before that which they do not know how to explain. Consciousness ceases its work, but leaves behind verified stories of people who visited there and returned. The body is destroyed, but becomes the foundation for hundreds of other life forms that exist precisely because it was there. Science in the 2020s stands before death approximately where medicine stood before sleep in the 19th century. The mechanism is clear, but the essence escapes. It is known what happens to neurons. It is known what bacteria do. It is known what waves an electroencephalograph records in the final seconds. It is not known if there is someone who experiences this. Science has not yet closed this question, and this is not a shortcoming of science. It is honesty in the face of data that do not fit into any of the ready-made schemes. Every second, about 2 people die on Earth. They depart into a process that has been studied more thoroughly than ever, and remains more of a mystery than ever. The border between life and death turned out to be not a point. It turned out to be a territory that is only beginning to be mapped.